So that was when I gave, that was 1978, and when we just completed our initial work and in the field of lymphocyte growth, T cell growth, and had identified an assay, made an assay for T cell growth factor and so forth. And one of the very first experiments that we did after that had already been done, really, in 1960 by a fellow by the name of Peter Knoll from the University of Pennsylvania. And he was the one that really, he made a breakthrough discovery in immunology, because he showed for the very first time that if you took lymphocytes out of the blood or the lymph nodes or wherever and put them into culture, there was a substance that you could add that was called phytohemagglutinin. Phyto meaning plant, hemagglutinin meaning that it clumped up red cells, red blood cells, and it was abbreviated PHA. If you put PHA in with your lymphocytes, and he discovered this by accident, and if you waited a couple of 3 or 4 days, that they would start to proliferate. And that up until that time, in textbooks, it was described, lymphocytes were described as sort of boring end-stage cells, and nobody really knew what they did, and nobody was really interested in them. Although they were in the lymph nodes, and when you get a sore throat and you have big glands in your neck, those are the lymph nodes. And so they're all over your body, and they circulate in the blood. And the fact that they could then proliferate was the thing that I started working on 10 years later in around 1970. And so I reread Peter Knoll's papers, and one of the first things he did was add steroids to these cultures. Not testosterone or estrogen or progesterone, the sex steroids, but rather the steroid. There's a whole cadre of steroid hormones that are made from the same sort of parent molecule, and the ones that he was interested in were called glucocorticoids, made by the adrenal gland, and they control the metabolism, or the way the body deals with all of the major foodstuffs: protein, fats, and carbohydrates. And carbohydrates and glucose, being the major sugar, was the thing that they thought these hormones were primarily regulating. So they had glucocorticoids, and it was discovered early on, back in the 50s, that these glucocorticoids, which are abbreviated corticosteroids, would inhibit the immune response, both, mostly in vivo in humans and animals, and also they were good for treatment of lymphocytic leukemia and lymphoma, and they would make the cells, die, go away. So the very first paper he did after he discovered the fact that he could use this PHA stuff to make the cells proliferate, he added glucocorticoids, and it stopped. So that was published in 1960. I didn't get into the game until 1970. And so after we had identified that there was this other stuff that we added the PHA, they started to proliferate, but before they proliferated, they made this T cell growth factor. So, it was obvious that the next experiment we had to do, we had to ask the question, do the glucocorticoids, do the corticosteroids inhibit that? So we did these experiments, and they did. And we could, with our assays that we had and so forth, we could pin it down to the fact that it stopped the cells from making the growth factor, the T cell growth factor. But if we added the growth factor and also the steroids at the same time, the cells were fine, and they proliferated just fine, so they could respond to the growth factor, but they couldn't make it. Well, that was the first light bulb. And that was a minor epiphany, and because I said, and this is where my cancer work came into play, and also at the same time at Dartmouth I had met a very interesting scientist who was an expert in glucocorticoid hormones. He was an endocrinologist. His lab was right upstairs from me. He was about 10 years older than I was. He was established because he had discovered the receptor for glucocorticoids about 10 years earlier. So we took our initial experiments up to him and we showed them to him and so forth, and we said, this is what we think is going on, and will you help us? And so his name was Alan Monk, and he was superb. And this was a very, very good relationship that started to then influence our work from there on after, because he was the fact that he was an endocrinologist, which means he studied hormones. He taught me the principles of endocrinology and hormones and what they are, how they work, and so forth. And of course, I'd already studied them in medical school and studied all the diseases that people get when they don't have enough hormone or where they have too much hormone and so forth. But that was really the beginning of the idea that it wasn't the initial stimulus, the PHA in this instance. It was the subsequent. It was the growth factor that was the key thing that made the whole thing happen. And that really is the critical discovery, because of the fact that then what you can do is you inhibit the production of the growth factor, or maybe you can find things to increase the production of the growth factor,
right,
or increase the response to the growth factor. And so that's then set the next series of experiment that went on for like 10 more years, essentially.
Wow.
And so that was and so we published that in 1979. And then we set out, at the same time we were purifying the growth factor and trying to identify the molecule, which we were able to do with Alan's help. And
this is IL-2 that you're...
This is IL-2, yeah, but this time it became IL-2. Which is another long story, which is a political story in the business of science. It's an interesting story too. So I did a lot of reading, and I'd already done a lot of reading when I had been in France, because we were working on a different hormone at the time that causes red cells to grow. And I knew that there weren't too many receptors for hormones that have been identified and discovered and characterized and so forth. There was insulin that enhances or allows sugar metabolism. There was nerve growth factor, and there was epidermal growth factor, and that was the only three that had been discovered in the late 70s. Oh. And so I read all those papers, and it was clear what we needed to do next, and that was that we needed to purify the IL-2, the growth factor. We needed to then radiolabel the growth factor so that we could trace it, because the next experiments then were to look for the receptor, and if a cell had a receptor, then the radiolabeled growth factor would bind to it, and then you could separate the free hormone from the cell-bound hormone, and you could then quantify the number of receptors and so forth and so on. And so Alan was just, he'd already done that with glucocorticoid hormones, and he knew exactly how we ought to do this, that, and the other thing. And so we did that. And so we published that in 1981. And after that, then it was clear to me, we did all of the critical experiments, and we got the data back. And I ran upstairs to Alan and I said, because it seemed to me that what we'd done in our experiments is that we'd shown that it satisfied all what were the classical characteristics of a hormone receptor interaction. And I ran upstairs to Alan and I said, Alan, see what we've got? This stuff is a hormone. And he looked at me and he said, well, because he was sort of grandfatherly. And he said, Kendall, I'm not so sure. If you haven't demonstrated negative feedback control, you don't have a real hormone.
Meaning you have to be able to turn it off.
Yeah. And all hormonal systems do that. They turn something on, but they also have a feedback negative loop that turns it off.
Right.
Otherwise, it would be just running willy-nilly all the time. And particularly with cell growth, you don't want them to just grow and grow and grow and grow. That's basically what cancer is.
Right.
So I thought about it. We did a series of experiments, as much as we could do, given the techniques that we had at the time, and we couldn't really satisfy ourselves that we could call it a hormone. But I did anyway. I published a series of review articles and so forth, discussing the whole thing and trying to really educate the immunologists that this was a hormonal system, because to me that was really, really important, because that meant that then we knew how to manipulate the system, because to manipulate the system we manipulated the hormone. We had the molecule discovered and identified and purified, and now all we needed to do was find ways to change that, either up or down, to modify how the immune system worked. And so I think that the thing about that whole process of me trying to educate the immunologists, they didn't like that. They didn't really buy that, because they didn't understand what a hormone was, and it was peripheral to what their interests were, because the rest of the field was involved in other kinds of questions, which was the good thing for me.
No one was looking where you were looking.
Yeah, exactly.
Right.
They weren't looking under the bush that I was looking under.
So what was the field of immunology's major, concern at the time?
At the time, it was basically not on this how the system was regulated, but how the system was activated, and via, in particular, the T cells as opposed to the B cells. And it was just around 1965 to 1970. Discoveries had been published and made that indicated that there were not just lymphocytes. There were at least two different kinds. There were B cells that came from the bone marrow and made antibodies. And antibodies were what all of the immunologists had studied for the past half century. So they were really focused on B cells and antibodies, but they found these other cells. They were in the lymph nodes and in the blood and in the thymus that ultimately we called T cells, but they didn't know what they did, and they didn't know how to deal with them. And the major problem is that they knew that if they added specific antigens to their cells, that then the cells would proliferate. But they didn't know what the receptor for the antigen was. On B cells, they knew it was surface antibody, and that had already been shown by 1970, and so that was good. And so they figured there must be a very similar molecule for the T cells, and everybody looked for it. They called it immunoglobulin T, but nobody could find it. And that was one aspect of things. And another aspect of things was the fact that it looked as though T cells only recognized antigens on the surface of other cells, which were called antigen-presenting cells, most of which were macrophages, the other cell of the immune system. So T cells couldn't just see molecules floating around. They had to have it on the surface of the cell.
Okay.
And people didn't understand why that was. And that was what everybody at the time that I was looking at trying to understand the regulation of the system. They were most of everybody else was focused on what activated the cells in the first place.
So things like outside forces acting on the body, triggering the immune.
Right, exactly. And so the whole idea was that, and the other thing was the dogma of the immunologists is that the immune system is special. It's different than all the other bodily systems, because by then people had worked out the fact that the cardiovascular system, which is comprised of the heart and the blood vessels and the blood, the cardiovascular system is regulated by the nervous system. There are nervous signals that go down to speed up or slow down the heart rate and so forth. And that had already been worked out, so that there were these ideas that the body's organ systems were regulated along with all the other organ systems. The digestive system as well, same thing. Nerves going to the stomach and the intestines were regulating that. But the immune system was billed by the immunologists as being distinct from all that. And it was only when an external antigen came in and then it activated the immune system, the cells proliferated, and then they dealt with whatever that antigen was and got rid of it in the body, and then they quieted back down again and became, resting, waiting for the next encounter. but the new thing and what the light bulb going on and the epiphany and the eureka is that wasn't true. And that there was an internal regulatory system that was regulated by the immune system itself that took over, sort of an endocrine system for the immune system. These hormones that were regulating everything. And if that was true, that means that what you needed to do is regulate the hormones and forget about this external business. I mean, it's still important.
Right.
And actually, one of my very, very best friends, ultimately, just a few years later, discovered the molecular structure of the whole T-cell antigen receptor.
Wow.
So he became a good friend of mine.
I should hope.
Yeah.
You'd want to join forces.
Well, there's a funny story that I like to tell, which is a true story. And I met him right around the same period of time that we're talking about, 78, 79, 80, around in there.
Okay.
And I had been invited to speak at a meeting, and so I got up and gave my talk, and then I sat down in the audience, and then other speakers would come up, and then this fellow came up. His name is Ellis Reinhertz, and he's at Harvard at the Dana-Farber Institute. And he gave his talk, and I said to myself, I'm sitting there listening to this guy, and I'm saying to myself, this guy's really smart. He's almost as smart as me. And I said, I'm not going to compete with this guy. I'm going to make him my friend. And so that's what I did. And we became fast friends, and still are. But then the other part of the story I like to tell is that then after I got to know him pretty well, I see he wasn't that smart at all. So anyway, yeah.
That's really. So the field was sort of focusing on. It was reactive more so.
Yeah.
Something outside coming in, the immune system reacts to.
Mm-hmm.
Whereas you're looking how to have the immune system be a proactive tool as opposed to a reactive.
Well, I wasn't actually at that time. I wasn't thinking along those lines, but I was thinking more along the lines that, this Peter Knoll guy that did the first experiments with PHA and published in 1960. I read his paper over and over again in 1970, and it was. The strange thing to me is that he would add the PHA on day 0 in the lab, but the cells wouldn't start to grow for 3 or 4 days.
So how did that happen? Because that was an accident that he discovered this.
Well, yeah. Well, the story that he told, and I've talked to him about it. He had some. He was he was a cytogeneticist. He was not an immunologist, and he wanted to get to the chromosomes of the white blood cells.
So what is a cytogeneticist?
It's what you do is you study the chromosomes of cells.
Okay.
And in order to do that, you have to sort of stop them so that you can look at them under the microscope.
Okay.
And there's a whole process to go about that. At the time, there still is, but now we've gone beyond that because now we study the DNA, the molecules themselves. Mm. And the chromosomes are great big collections of genes made out of DNA. So I said to myself, well, why does it take so long? Why don't they just, I can see how they take 24 hours, but they take 3 or 4 days?
Gotta wait a week each time you want to do something.
And he discussed that in his paper. And he said, perhaps the cells are making some other factor that makes these cells grow. So he said that in 1960. And so when we found what that other factor was, or we thought we did, it all made sense.